Our next speaker is Stephanie C. She is president and CEO and founded Meditope Biosciences, which is based on some technology out of the City of Hope. She has 20 years in the biopharma industry. She is also a patent attorney. Prior to this, she worked in Impax Laboratories. She has a degree from Wesley College, an MBA from Stanford, and a JD from Columbia University. And she's going to tell you about her antibody-based cancer products. Stephanie?
I think we're the youngest company here, so this will be short. I'm also probably the least technical person up here. So as you can see, I come at it more from a legal and business background. So my sweet spot has really always been at the intersection of the regulatory, intellectual property, and new product planning strategy intersection, I guess. So we are, as you see, just over 2 years old. We were founded in May of 2012. We raised our first round of financing just over a year ago. It was a $3.6 million, basically a seed round. So it was all raised from private individuals, mostly based here in the LA area. So to the extent that people aren't, our investor base isn't here, they were definitely sourced through our LA investors. So there, Ahmed, there's a strong source of funders and people interested in getting involved in this area here in LA. One thing I'd like to add too about our pool of investors, it's a really nice diverse mix and a healthy mix of backgrounds and industry experiences. So we've got people from the industry, we've got scientists who I would describe, they'd probably hate hearing me describe them as luminaries and pioneers in antibody engineering. We've got private equity guys who've invested just their personal money, as well as just a lot of people from all walks of business life in different industries. And it's made the conversation over the last couple of years, as we've gone through this process of starting the company and working our products through and working our technology, it's made the conversations really rich for me anyway. So the technology itself comes from City of Hope. It's a platform technology that we licensed from them. It's, in short, we like to describe it as turning monoclonal antibodies into a Lego system. So what it does is allows one to pretty much snap on or snap off very easily and very elegantly and very consistently just about anything imaginable to a monoclonal antibody. It's essentially a two-piece system, so what we do is we kind of sculpt or graft a site into the fab of the monoclonal antibody, and then on the second part of it, we have a series of small peptides. You could use small molecules as well that plug in specifically to that sculpted keyhole, if you will. And so with that, you can conjugate directly onto this very much more manageable and manipulatable peptide that then locks into the antibody. And we've done this with several antibodies now, and there's no effect on the way the native antibody behaves. So it still finds its antigen with the same specificity and binds with the same affinity with the changes in it, as well as with the Meditope locked in there. So what else can I tell you about us? That's the technology. We are based in Pasadena. Our corporate headquarters are in Pasadena, but my R&D team is basically in San Diego, so we're somewhere in between LA and San Diego most of the time. And we basically spent the past year really just working with the technology platform that was handed over to us from City of Hope, validating some of those results and really gaining a better understanding of what we could do with this platform. I think when we first took it over, we could see a myriad of applications. I mean, anywhere antibodies are used, there are implications for this technology. And so we kind of said at the beginning that, we wanted to go for antibody drug conjugates, and that we thought that that was a great place for us to be for a number of reasons. It being a hot area in pharma right now, as well as the fact that we thought that the benefits of this platform technology would really be showcased really nicely in that arena. So this past year was really about getting to understand if this platform could be used, and if we could get there in a practical and efficient way for developing antibody-drug conjugates. So we're right on the cusp of sort of coming out of that tech validation research stage and starting to develop our own ADCs right now.
Great, thank you.